Alrighty guys, so we're going to start chapter 12 in the Pierce text, and this is the first of the next 3 chapters that are going to be really heavy content material. There's going to be lots of moving parts, and it's kind of hard to visualize these concepts sometimes, so I encourage you to spend a lot of additional time studying. In addition, this chapter is going to be the last in the second set of chapters that will be on exam number 2. So after you get through this material, you should start prepping for the exam. Alrighty, so chapter 12 is all about how we replicate DNA and how DNA can go through the recombination process. And it's a pretty big chapter with lots of little sections, so we're going to hit it in kind of in this order. So give you an overview of the process, and then we'll talk about modes of replication in this link, and then in following links we'll get into requirements of replication, directions of replication, how replication actually occurs in bacteria versus eukaryotic systems as well. Alright, so when we talk about replicating DNA, this is actually the process of taking a DNA strand and copying it into another DNA strand for the process of meiosis or mitosis. So this is happening during the S phase. And some of the requirements of this DNA is that in order for replication to happen, it must be very accurate and very fast. So for example, E. coli has about 4.6 million base pairs in its entire genome, and it has the ability to copy and divide every 20 minutes. So that's pretty astounding. DNA on average is replicated in E. coli systems at a rate of about 1,000 nucleotides per second, and it very awesomely has only fewer than 1 in a billion errors. So that's a really high accuracy rate. So replication is conducted in a semi-conservative manner, meaning that the original nucleotide remains intact and is used as a template, so it's conserved. So we're going to talk about 3 modes of replication. The first one is theta replication, and this happens in circular DNA. And theta replication is unique because it can happen in a bidirectional pattern. So what we're looking at here in this slide is up here at the top is a micrograph of an actual cell dividing, and we're going to look at it more in depth on the next slide. But the process is really diagrammed very well for you down here at the bottom. So in bacterial systems, we have a circular chromosome here, and that is double-stranded DNA, and it is going to unwind at the origin of replication, which is shown here by this little yellow tick mark. So after that unwinding starts to happen, then that is going to produce a single-strand template for the synthesis of new DNA. So the newly synthesized DNA is shown in red here, and the template or the original strand is shown in gray. And so this that forms right here after we start building that new DNA strand is actually a replication bubble. So that bubble is going to form and is going to have a replication fork at each end. And we can see here that the replication fork looks like this. That's where we're actually starting to synthesize the new piece of DNA using the old original pieces of DNA as a template. So with bidirectional replication in the theta model, we then are going to see forks that are going to expand here in step 3 and get bigger. So our replication bubble gets bigger and our newly synthesized DNA gets a lot bigger as well, until we actually have 2 circular DNA molecules that are produced. So those circular DNA models includes one of the old strand or the original strand of DNA and one of the newly synthesized strand. So in conclusion, the products of theta replication are 2 circular DNA molecules. So when we look at what that looks like in the cell, this is an electron micrograph. So we've got the chromosome is actually shown here in a circle, and we can see that we have the start of the replication bubble forming and replication forks on either end, and right here in the center is the origin of replication. So as that theta replication process continues to progress, we start getting theta replication occurring, and that replication bubble gets a lot bigger, and eventually we get two newly synthesized pieces of DNA that create two cells. So this again is happening in the S phase. The second model of replication is the rolling circle replication. It also occurs in bacterial models and smaller genomes. Okay, so the rolling circle replication is going to be a little bit different than theta because it's not bidirectional, it's unidirectional, meaning that it only goes in one way. So where we have a double-stranded piece of DNA or double-stranded chromosome here in a very simplistic cell like a bacterial cell, if that cell undergoes rolling circle replication, then the replication is going to be initiated by a break in one of the nucleotide strands. So that break that we see is actually going to be on that outside strand. And again, these are oriented 5' to 3' with that additional OH group and the phosphate group helping that process. So as that break begins to become more prominent, DNA synthesis is then going to begin at the 3' end of the broken strand. Okay, so DNA synthesis is going to begin at the 3' end of the broken strand. And then the inner strand is actually used as the template or the original strand to make the copy from. And finally, the 5' end of the broken strand is going to be displaced. It's just kind of hanging out in the cell of these bacterial organisms. And so as we start to add more and more nucleotides to create a longer and longer newly synthesized strand, as we can see here, or shown here by the red, then we end up getting a cleavage site that is going to release the single strand linear DNA and a double-stranded circular DNA, so that the result actually result in 4 different products. So the linear DNA is going to be clipped off of the original template, and it may be used later to serve as a template of synthesis for another piece of DNA. This is one of the reasons that bacteria can repopulate so quickly. And then the other product is going to be this newly strand of DNA, which can then serve as a template for another new strand of DNA. And then we have the original cell, which has one of the original strands of DNA plus the newly synthesized strand of DNA. So one thing that is important and where we get our 4th product from is this newly synthesized strand actually has the ability to go through the cycle again and be repeated as well. So that is rolling circle replication. Again, it still occurs in bacterial models, but it is definitely a different mode of replication compared to theta. All right, so this is just looking at that process a little bit more in depth. So we've got the original template strands being cleaved at the origin. Our 3' end begins DNA synthesis. Our 5' end is going to be displaced. We add more and more nucleotides to the newly synthesized strand, and we get one of 4 products. Okay, so these are going to be these products are going to be multiple circular DNA molecules. And finally, the third mode of replication is linear replication. That usually happens in eukaryotic cells because chromosomes in eukaryotic cells tend to be linear. So one big difference is that in eukaryotic systems on linear chromosomes have multiple sites of origin. So it's not just like theta and rolling circle where there's one site of origin. There's actually multiple sites of origin within that chromosome. So in eukaryotic systems, because there's so much more genetic information to copy, the replication is actually going to proceed at 5,000 nucleotides per minute. And this is really, in essence, pretty slow, especially compared to like transcription translation. So it would, if we only had one site of replication, it would take 7 days to replicate an entire chromosome at this rate. So that's why you need multiple sites of origin. So replication in the eukaryotic model looks a lot like this. So we've got our original linear chromosome up here at the top, and then we've got a one site of origin that is starting to form a replication bubble here, another site of origin that's going to form a replication bubble here. And as they get longer and we start adding more nucleotides to that newly synthesized strand on both of them, they eventually meet and are glued back together to form this long piece of DNA. And so at the end of this process, all the forks are going to fuse together, and we have an entire new copy of a chromosome. So again, this is happening in a bidirectional manner. Okay, so similar to theta replication, but in a linear model. So when we look at the number of origin sites, it varies a lot depending on how complex the genome is. So small genomes such as E. coli only has one site of replication, but large genomes like the mouse and the frog, they have multiple sites of origin. Okay, so the average replicon, or the average length of a replication, is going to be very long in E. coli, and it's going to actually be shorter in the more complex systems. All right, so this this table in your book really gives you a good idea of the different models of replication, so theta, rolling circle, and linear, what DNA template they're going to be specific to, and then whether they are going to have multiple or only one replicons as their products, and then the products of each one of those models as well. So this gives you a really good overview of what we're headed into in DNA replication. So we're going to be looking at how each one of these processes occurs at a more detailed level.